Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled The Added Value of Biostatistics-Driven Randomization and Trial Supply Management System. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario delivers the leading endpoint technology solutions for clinical trials. Through experience gained from over 19,000 clinical trials delivered in support of 870 regulatory approvals, Clario fuses scientific expertise and global scale into the broadest endpoint technology platform to enable pharmaceutical, biotech, and medical device partners to transform lives. Through Trial Anywhere, Clario has mastered the ability to generate rich evidence across all trial models, decentralized, hybrid, and site-based clinical trials. Now I would like to introduce our speakers for today's event. Paul van Basterhout is the Senior RTSM System Specialist at Iddi, a therapy-focused expert clinical data services and high-level strategic consulting contract research organization committed to increasing efficiency and mitigating risks through the drug development process. He is an expert in randomization and trial supply management from trial design to data submission and is dedicated to maximizing business results. Paul holds a master's degree of science in mathematics. And Tony Warren is the product owner of the Clario IRT and Trial Supply Optimization (TSO) software solutions. Tony became involved in IRT during the days of phone-based IVR systems at a CRO, Kendall International, back in 2000 of January. Tony led the growth of this product and its transition to a hybrid phone and web system, as well as the development of advanced algorithms for dynamic minimization and clinical supply management. Tony joined Clario in 2021 and has led the expansion of the Clario IRT system to support direct-to-patient program trials, clinical supply forecasting optimization, and more advanced dynamic minimization algorithms. But without further ado, I'd like to hand the presentation over to our first speaker for today, Paul van Basterhout. You may begin when you're ready.
Thank you, Ryan, and thank you for this nice introduction. And sorry for my sound issue. So let's start with the agenda for today. There are a few points that we would like to cover during this webinar. The first point is a quick overview of what we mean by biostatistics-driven RTSM. We will then cover 3 case studies where statistical expertise and RTSM reconfiguration were essential for the study's success. And the last point, we'll revisit the 3rd case study, and it will enlighten and bring a new eye to trial supply optimization. But before we cover these points, we'll have a little poll for you, and I will let Ryan introduce it.
Thank you very much, Paul. So appearing on everyone's screen right now is a polling question that they can interact with in real time. Please select one of the answers you see in front of you and then click Submit to participate. The question that we're asking is, have you used minimization in clinical trials? Your answer options are yes, no, or unsure. I'll give everyone a few seconds to consider your answer to the question again of, have you used minimization in clinical trials? Looks like most of you have submitted your answer, so thank you very much. Let's take a look at where your results have come in. We have 45% for yes, then 38% for no, and 17% of you selecting unsure. So again, thank you so much for participating. Before we continue, though, I believe we have a second poll question that we'd be really interested to know your opinion on. Now, our question is, are you handling your randomization strategy in-house or using external consultancy? Your answer options are in-house, external consultancy, in-house and external consultancy, or again, unsure? Everyone, a few seconds for this question as well. In a moment, we'll hear more from our speaker, Paul. Again, the question, though, is, are you handling your randomization strategy in-house or using external consultancy? We have the majority of answers in. Let's look at your results. We have 57% of you selecting in-house, then 23% for both in-house and external consultancy, 11% for external, and then 9% of you saying unsure. So again, thank you so much for your participation. Paul, it's back to you.
Thank you, Ryan. These are indeed interesting results, and I want to thank the audience to be so reactive. We will be covering some aspects of minimization in the case studies. And regarding the in-house versus external statistical consultancy, I would like to mention that part of the IDDI core services, we do provide clinical, statistical, and regulatory consultancy services, helping sponsors write protocols, define their randomization strategy, and more. And to pursue on this short IDDI introduction, as you may guess from this seminar agenda, we also offer integrated solution services, working with technology partners like Clario for the underlying RTSM and EDC solutions, but setting up, validating, and supporting those services and integrations for the client all over the study lifecycle. Data management is another of our core services that focuses on the accuracy of the clinical data. Our clinical data management experts are committed to delivering timely, accurate, and high-quality clinical data. Last but not least, the biostatistics services. That is IDDI's DNA. These are the services that the company was built upon over 30 years ago. These core competencies, combined with Clario RTSM know-how, allow us to deliver what we call a biostatistic-driven RTSM. Together, we ensure that your studies are configured in the most reliable and efficient way. In short, we are there to secure the trial integrity. Our biostats teams actually intervenes at every step of a study, from study design to setup and during the full study follow-up. They play, of course, an important role at the study closure, but that concerns more the clinical data management services. For the RTSM, at study design, our biostats teams help defining the randomization method with the sponsor for the study. They define the stratification factors for the study, together with the medical expert, may advise on sample sizes, and they run simulations to see the subject distribution per stratification factor, ensuring that the best randomization method is chosen. For some studies, this study design consultancy is where our services ends, actually. We are just providing that statistical consultancy. When we move to the actual implementation of these designs, the biostats team intervenes again at the study setup, performing a revalidation of the randomization algorithm and checking kit allocations to ensure that the correct drug with the correct dose was allocated based on the assigned treatment group and the defined schedule as specified in the protocol. Once the setup is approved and we move to production, the biostats teams continue to do monthly check to ensure the kits allocation are done accurately and the randomization is well balanced. But let's move now to the first case study. In the previous slide, I highlighted the usual interventions of our biostats team during the study lifecycle. But in some cases, some unexpected situation may come up that require advice and maybe mid-study reconfigurations. That is the case of the two case studies that I will describe in the coming slide. This is the first one, where we were requested to adapt the minimization algorithm. First, a few words on the background. This was a phase 3 study. The study was evaluating the efficacy and safety of an active drug, comparing treatment results against placebo, and the randomization method was used was minimization. Minimization is a dynamic randomization method that calculates for each new randomized subject the treatment group that minimizes the imbalance for its stratification criteria. The importance here is the dynamic aspect. as the treatment group is calculated for each new randomized subject. This, compared to a permitted block or a randomization list that is important, that is static, and where the treatment groups are actually predefined in the list. Here, the minimization ratio is 2:1. So we would expect to have 2 times more active patients than placebo in the final subject review. The challenge with this study, as you can see in the treatment, is that the patients were randomized, and when randomized to the treatment group, only then was the product actually manufactured using the patient bone marrow. The drug used could sometimes not be manufactured because maybe there was not enough material to do it, or the manufactured drug did not match the acceptance criteria. These patients were therefore not treated, which somehow skewed the active subject balance. And the sponsor asked us to propose a solution. The IDDI Biostats team and the RTSM specialists sat together and evaluated the situation with the sponsor. The proposed outcome was to update the randomization design in the RTSM system. The new design now dynamically excluded the untreated and discontinued patients. And this was only possible because we choose the randomization method as minimization. So if we look in the initial design, where all the patients were included, we see that we have 1, 2, 3, 4, 5 active patients and 3 placebo patients, which means that the active treatment group would favor the balance. We have 5:3. If we add an active, we would have 6:3, which has the 2:1 ratio, which match the 2:1 ratio. With the new design, where we excluded the not treated and not continued patients, now we have a situation where we have 1, 2, 3, 4 active patients and only 1 placebo patient that are included. And so in this case, the placebo would favor the balance. We have 4 active, 1 placebo. The placebo, adding 1 placebo would do 4:2, which would favor the balance. So the sponsor approved this new approach, and the mid-study change was implemented. The second case study is another quite interesting mid-study change required by the sponsor. Again, let me first say a few words on the study background. So this is a phase 3 study. The study was comparing the efficacy of 2 drugs, and the randomization method again was used was minimization again, with 6 stratification factors. The number of stratification factors already lean to the minimization method, but we will see that it played an important role in this study change. The sample size was relatively large, about 700 patients, 700 subjects. And when the sponsor checked an intermediate results, as 187 patients were randomized, he identified an important biomarker that could impact the study outcome. This biomarker was actually identified because of 10 events that were related to it, and they saw that in total 27 patients amongst the 187 so far had that biomarker. More importantly, while the overall balance was as expected, 94 drug 1, 93 drug 2, when we look to these 27 patients with that biomarker, we saw that the distribution was unbalanced because 10 were in drug 1 and 17 were in drug 2. So the sponsor requested actually that we integrated that biomarker as a stratification factor. We could either replace an existing factor or add it to the system, but this was an important element that he wanted to randomize around. So again, the biostats team and the RTSM specialists sat together and evaluated the situation of the sponsor. The biostats team advised to add this important biomarker as a stratification factor. On the other hand, the RTSM team was able to import the collected data for this biomarker for existing subjects. Hopefully, the sponsor had collected that information and we could reuse it. As a consequence, the overall balance among the two arms was reached for the 7 stratification factors. I would like to go a little bit more in detail in this in this case study, just to explain the different the situation. So this was the initial situation, as you can see, with our 187 patients stratified. Obviously, these numbers are just here for illustration, but stratified and then assigned to the different arms, drug 1 and drug 2 arm. If we had just added a 7th stratification factor, what would have happened is that the minimization calculation would have started again with the patient 188 because we didn't have any information about the stratification factors before. But as the RTSM team worked and was able to integrate and add these previously collected values, we were able to actually continue the minimization calculation as before and provide at the end an overall balance. So this assured an overall balance over time for the two arms considering the 7 stratification factors as requested by the sponsor. A last case study here does not relate to randomization methods, but as you will see, collaboration between our biostats and RTSM team goes beyond that. Here the challenge consisted in optimizing the drug supply for an expensive drug. Again, first a few words on the study background. This is a phase 2 study. The study was assessing the efficacy of an active drug versus placebo, and the minimization method that was used here is of less importance because we are more focusing in this case on the drug supply itself. The challenge here came from the fact that the drug was expensive to manufacture. Add to that the fact that the drug was dispensed in different dosages, 5 milligram vials, 3 milligram vials, 2 milligram vials. In case of an event, like some of the intolerance, for instance, the drug dosage was reduced by 1 level. And of course, it was difficult because it's event-driven to predict the quantity of 3 milligram vials that were needed and the quantity of 2 milligram vials needed even more so because these 2 milligram were depending on the move from 5 to 3 and the move from 3 to 2. To add to the complexity, the sponsor used central pharmacies. You probably guessed it already, the request from the sponsor was to optimize the drug delivery and find a way to make sure we shipped as little drug as we could, yet ensuring sufficient vials were on site for the upcoming patients. This was again a collaboration effort between RTSM and the biostats team. The RTSM team built a predictive drug supply model, introducing variables, percentages, that could be filled later by the biostats team. The model is schematized here in this slide. On the left side, we have the requirements for the initial shipment, and on the other side, we have the requirements for the trigger and the resupply values. So the quantities, when we need to order new vials or new drug, that's the trigger, and the resupply value, how much do we need to ship to that site. When we look to the initial shipment, this was agreed upon with the sponsor that we would have 6 kits per central pharmacy site. So if the central pharmacy was serving 3 sites, we would have 18 kits for this pharmacy. And we decided to have for 3 milligram a percentage of the 5 milligram quantity, and for the 2 milligram vials another percentage of the 5 milligram quantity. For the trigger and resupply, we also add some calculation. The trigger it was decided for the trigger together with the sponsor that we wanted to make sure that we had sufficient drug on site for the next 2 allocations. So if the number was below this required quantity, the resupply would start. And the resupply value was calculated to actually fill the gap between what we have now on stock and the next 4 vial allocations. So trigger, I need to have sufficient vials for the next 2 vial allocation. Resupply is made so that I'm sure I have enough vials on site for the next 4 vial allocations. This then turned into a number of 5 milligram subject in all, that was just based on the number of 5 milligram subjects. We did add some buffer. For the number of 3 milligram subjects we took for 3 milligram vials, we considered, of course, the subjects that were already on 3 milligram, plus we added a percentage of the subjects that were on 5 milligram going down to 3 milligram. And same for the 2 milligram, we considered the subjects that were already at 2 milligram and added on that a percentage of the 3 milligram subjects that would go to 2 milligram. And all these percentages were analyzed and advised by the Biostats team. And they could, of course, change over the course of the study. The Biostats team could review the last information provided, the last trends, and the RTSM team could re-implement the new values advised by the Biostats team. So that covers my part. As you have seen, the predictive supply model used in the last case study was using past data to optimize the shipped drug quantities. It would be nice also if we could predict those behaviors before the trial even starts and adapt it as the trial goes on. And that is the part that Tony will cover now. So Tony, I'm passing you the word.
Okay, great. Yeah, thanks, Paul, for that transition there. And yes, what I'm going to discuss is a capability within our IRT system for predicting what will happen in the study. This is a new dimension that we're adding to IRT, which traditionally allows you to look at what has happened and where you currently are in the trial. But the optimization and forecasting capabilities allow study managers and clinical supplies managers to have a look into what is expected to happen based on current trends and assumptions, and then forecast and optimize your trial supply accordingly. Next slide, please. So we will have a poll question, Ryan, if you want to take that.
Yes, thank you so much. So appearing on everyone's screen is another polling question. This one is wondering, do you have experience with supply optimization tools? Your answer options are simply a yes or a no. The question again, do you have experience with supply optimization tools? And you're all very fast at answering this one, so thank you so much for participating. Let's go ahead and take a look at your results. We have here 80% of you selecting no, and then 20% of you selecting yes. So again, thank you so much for participating, and I'll hand it back to you, Tony.
That's great. So I am pleased to be able to give you an overview of what we mean by supply optimization and how it works and integrates with the IRT system. Now, Clario has been providing a standalone trial supply optimization tool for a number of years now. And the way it works is you configure the system just like you do an IRT for the parameters of the study, the randomization strategy, treatment groups, drug types, visit schedules, etc. And then along with predictions on the number of sites and the enrollment rates, we run a Monte Carlo simulation of the study, which Monte Carlo just meaning we use random numbers around the probability of various events. And that allows us to simulate activity over time, when the rate at which subjects get screened, the rate that they drop, randomized, et cetera. So we have a forecast of what's going to happen over time with the trial. And then that forecast can be used to determine the kit demand at each site over time. So we get a time series now of the predicted demand. And from that, we can use that for planning how much study drug is going to be needed, et cetera. Now, as we all know, the assumptions that we make at the start of the trial will change over time. Either enrollment rates will be faster or slower. We may need to add more countries or sites. There may be mid-study changes that need to take place. And just some of the assumptions, such as the screen failure rate or the randomized subject participation rate or the dropout rate. And if to add additional complexity, we can have a study where there's variable dosing, such as Paul's case study 3, where we have the option to titrate the drug dose at various visits. So we have all these different factors that are playing in. We make our initial assumptions, but what we want to know is, as the study unfolds and these events occur, how will that change what our forecast is? Are we still on target? Is the study going to take longer? And if the study is going to take longer, what's the impact of that on clinical supply? And how much drug do we need to be supplying to sites? We certainly don't want to oversupply sites or undersupply them and risk a subject not being made of the drug not being made available. And even the amount of drug that we provide to sites at any given time is going to vary. So initially we may want to have additional drug available, and then as the enrollment rates for that site begin to taper off, we can stock them with less drug and still ensure that all subjects have the required drug that's available. So we can combine the data the actual data that's available in the IRT with our forecasting model and display that in forward-looking reports and optimal resupply settings as the study progresses. So let's take a look at this in detail now. On the next slide, please. So again, this is just an overview of that study that Paul discussed with active and placebo treatment groups randomized 1-to-1, but within the active arm, subjects will be dispensed a 5 milligram kit at randomization, and then at subsequent visits, the 5 milligram patients can step down to 3, the 3 milligram patients can step down to 2, and the 2 milligram patients remain on 2 milligrams. So there's variable dosing that's going to take place just based on the subject's tolerance of the study drug. Placebo patients will remain on placebo throughout the trial. Within the IRT, though, the investigator will be given the option to titrate them since they're unaware that which treatment arm is on. So we do that to maintain the blind, although there's no dose adjustment if they elect to titrate. Okay, next slide, please. So some additional assumptions. We're going to assume that there's 20 sites, and these sites are not all coming up at once. They're going to come up with a variable model, and we're going to have a screening cap of 300 subjects. So that will determine the completion of the study once we have 300 screened patients. And then, so the visit schedule is going to be, visit 1 would be screening, visit 2 would be randomization and initial drug dispensing, and then we'll have regular drug dispensing visits at intervals from visits 3 to 7. Now, subjects could, another variable that we need to take into account is subjects could come in at unscheduled visits in between for either a kit replacement, so they've lost a kit or the kit's been damaged, or they're having some response unexpected, and there may be a need to titrate that patient to a lower dose level. So all of that could occur at scheduled visits or unscheduled visits. And then visit 8 is the completion visit. Next, please. So one of the things that we want to take into account is what is the expected enrollment rate? And this, of course, is an unknown sometimes, but we make our best estimates. And we may have a standard rate that we want to use, like the one I have on the right, but there may be a site or sites that are higher enrollers. And we can set these enrollment rates within our model, either at the site level, or we can group sites into certain groups, such as a group of high enrolling sites and low enrolling sites, and apply enrollment rates based on that classification. It can be done at the country level. So there's really a lot of options. You can be as generic or as specific as you need to be based on your confidence in these estimates. So in this example, I'm just going to use two rates, one for our high enrolling site, Site 101, where we'll start out at 4 subjects per month, and the horizontal axis is months from site activation. So because sites are going to have a staggered activity period when they come on board. So we may only start the study with 2 or 3 sites active, and then over time, the additional sites come on board. And that, of course, is going to play a big factor in our modeling of subject activity over time. So again, 4 subjects per month for the first 4 months at our high enroller, then that drops to 2, and then it sort of tapers off to 1, as you can see. And then our other sites, we're going to assume a slower ramp up with just 1 subject per month initially, and then to 2, and then we'll peak at 3, and then start to taper off again. And again, we make these initial assumptions, and then as the study progresses, we can change those assumptions based on what we now think it will be, and then take a look at what the forecast is based on our most recent data. Okay, next, please. So there's some other things that we'll need to take into account, such as the screen failure rate, which I'm going to assume is going to be 20%. The post-randomization dropout rate, I'm going to assume for this simulation a 10% post-randomization dropout rate. I could refine that in more detail by having predicting at which visit the dropouts would occur. So I might say after visit 2, 1% of the patients would drop. At visit 3, 2%. However, again, I can be as detailed or as generic or as general as I think I can be with my assumptions. And I can also change the dropout rate based on the treatment arm that the subjects are randomized to if I thought that the active subjects maybe have a higher dropout rate than placebo or vice versa. But in this case, I'm going to just assume a 10% rate, at least initially. Can always change that as the study goes on. The assumption 3 is going to be the probability of unplanned visits. So these are the number of unscheduled visits between any two scheduled visits. And in this case, I'm going to assume that because of the titration and there may be some need to down titrate subjects, I'm going to assume that the active patients are going to have more frequent unplanned visits than the placebo ones. Placebo ones would be primarily for a kit replacement, but it's, they could be having some kind of unrelated response that the investigator wants to down titrate for, since the investigator is blinded. And then when we do have a visit, what's the probability of a dose adjustment? So as you can see here, we're assuming that over time, the 5 milligram patients will have a higher probability of titrating to the 3 milligram dose. And then similarly, the rate that 3 milligram patients down titrate to 2, and of course the 2 milligram patients remain at that level. So these are the assumptions that we make initially. And again, as the study progresses, we can make these adjustments to help fine tune the forecast. Okay, next slide. So as I mentioned, so even before we start talking about optimization, we're going to run the forecast of the trial using the assumptions I just presented on the time series, and take a look at what the subject activity is going to be like over the based on those assumptions. And again, we have a screen cap of 300 subjects. So we can take a look at this graph here, and we have a nice time series forecast. The gold bars on the far left of each month box represent the number of screen patients. So you can see the study starting in January, we would have roughly 15 patients screened this month, and then you can see the trend over time with a peak in May of 2023, and then we start to drop off with the last patient screened in August of 2023. And again, this is data that you can log into the IRT, and whoever has permission to have this view can pull it up and see the most recent forecast. And as actuals come in, the forecast will be rerun, and the actuals will serve as the initialization point. So if January ends up having higher enrollment or higher screening than expected, the forecast would be adjusted accordingly. And if it's lower, same thing. The green bars represent the subjects that have screen failed. Blue, the number of randomized subjects by month. Pink are the randomized subjects who drop, and then the orange bars are our completer. So based on our current forecast here, we can see that the last patient randomized is forecast for September of 2023, and the subject will complete in December of 2023. So nice visualization on where we're at with the trial with our subject activity. Now on the next slide, if you don't like looking at increments per month, but are like raw numbers and want to see the cumulatives, so this is another view that's available. So we can see at any one time, like let's say we look at June of 2023, by that point we expect to have 231 screened, 45 screen drops, 161 randomized, et cetera. You can see we cap out at 300 in August. And at the end of the study, based on these assumptions, we expect to have 238 randomized subjects and 216 completers. So, okay, next slide. So based on that level of subject activity and the visits that will occur over time, this graph gives us a forecast of the kit consumption. So these are the kits that actually get dispensed to subjects during that time series. Again, we start off slow, as you would expect in January, since there's not much left this month, with just placebo and active kits being needed, and same for February. So we have we only need those kits. So if we had, for example, this would be interesting, if we had, for example, a limited supply of 3 and 2 milligram kits, you know, we might have enough time to receive the next lot, because according to the forecast here, we're not going to even need the 2 milligram kits until April of 2023. So this gives you, for this type of trial, gives you an example of the demand for some of the kits and maybe some additional buffer that you have for getting those kits to the sites. And again, since half the patients are randomized to placebo and all placebo subjects remain on placebo, you can see the pink bars, which represent the placebo demand. You would expect that to be the greatest. And again, the kit consumption would be tapering off by December of 2023. So on the next slide, just like before, we can take a look at the cumulative demand over time that's forecast, based on our assumptions. And we can see the kits that we're going to need to be, again, this is the kits that we need to dispense to subjects over time, as well as at the far right then would be the total number of kits that were dispensed to reach the end of the trial. So now we have a forecast of how many kits are we actually going to be dispensing during that time. Now, again, the number of kits required is going to be higher than this, of course, because we have to ship kits to the sites because there's uncertainty with the titrations and uncertainty with the randomization. So there will be a bit of an oversupply. So how many kits are we going to actually need to ship to sites? And that's the next view on the next slide here. So in order to meet that demand and minimize the risk of run out where kits that are required by the subject are not available to site, this is the demand. And as you would expect, early on we would be shipping a lot more of the active and placebo kits to have sites, those kits in place at the sites to meet that demand. And then over time we'd be shipping more of the 2 and 3 milligram kits. Of course, the placebo demand is going to be fairly constant until the subjects begin to taper out of the trial, or all complete or drop from the trial. And then again, you can see again the demand, or the actual amount that we need to ship to sites over that time. And again, the next slide gives the cumulative numbers. So this is a great view for the clinical supplies manager. These are the kits that you actually are going to need over time to be shippable to the sites. And in the far right column, you would see the total demand that you would need. So right away, I know exactly the forecast of over time as well as total. So I'm going to need 756 placebo kits, 507 5 milligram kits, 268 3 milligram, and 119 2 milligram kits to support this study. On the next slide, please. So that's all great information. How do we apply it, and how do we make use of it? So this is a screen from my IRT, and this is a fairly standard way of configuring resupply settings for sites. How much drug do I need to initially ship to sites? What's the floor quantity? In other words, what's the level of inventory at the site that triggers a resupply? And the ceiling quantity is how much do I need to bring that inventory back to that level? So there's various ways that clinical supplies managers determine that now. Some have spreadsheets, some have various just instinctive ways of doing it, but this can be a difficult thing to manage, especially in a trial where there's a lot of moving parts. So we make our best, in the past we make our best estimate, and we can also indicate, just as an aside here, when we want to ship those kits to the site. So we can make it at site activation, or we can make it at a specific visit. But what really should those values be? And again, they're going to be time dependent as well too. So whatever we set initially, a month from now, we may want to adjust those values because the low enrolling sites may need less and the high enrolling sites may need more as subjects start coming in. So it's important to remember that there is going to be a time dimension to these parameters that you want to take into account. So they should always be reviewed periodically for accuracy. But what our simulation does now that we have forecast the demand of kits over time, we can then make a determination of what the optimum values are to balance oversupply of sites versus and having drug that's wasted versus making sure that we have sufficient drug to meet all the upcoming patient requirements. So if we go to the next slide. So this is the what the IRT then will also, this is the clinical supplies manager, in addition to being able to view the views on the forecast of subject activity and kit requirements, we'll be able to see what the system is recommending as the optimum values currently for those sites. So we can see here that the system is going to recommend that we have an initial, I had estimated setting 6 5 milligram kits, and of course we do this by kit type. So the upper one is 5 milligrams, and just a reminder, we had one site 101, which was a high enrolling site, and 102 and all the rest are going to have the same values because they're based on the same set of assumptions. So our high enrolling site, it's recommending that we can get by with 5 as the initial kit, so one less kit. And then for the low enrolling site, instead of having 6, it's saying that we only need 3 kits to ship initially to the sites. And again, that decrease of 2, if you have a lot of sites, in this case we only have 19, but some studies have a lot more sites, adjustment of 1 or 2 can have a significant impact in cost as well as in the number of kits required to support that. So here we can get by with 3 to optimize the resupply that's available. As far as the floor or when we trigger a new shipment, again, it's going to recommend that we stay with our value of 3 for the high enrolling site, but for the low enrolling sites, we can adjust that down to 2. And similarly, we can see what the values are for the ceiling floor. So based on what I had initially estimated, we can get by with lower numbers and still not run the risk of running out of drug. Now, the IRT also has a predictive resupply algorithm so that once subjects are randomized, it will make an estimate as to what that demand is going to be needed as well, so that if a site does have an imbalance of active or placebo subjects, that will be taken into account as well. But these are the settings that that are recommended that the clinical supply manager apply to optimize resupply. And on the bottom graph, we have the case for the placebo drug. Now, it's interesting here that there's no distinction between the high and lower enrolling sites. And the reason there is because all placebo patients are going, and that's half of them, are going to stay on placebo throughout the trial. So there's no risk involved in supplying a full 5 kits because they will be consumed by those patients over time. So those are the recommended values there. So here you can see that, number one, we get a lot of data that's forward-looking, which is a new dimension to IRT reporting, which traditionally has focused on what has happened and where we're currently at. It adjusts over time, so we make use of the actual data to improve that forecast, as well as making any other changes. Now, there is an integration between the IRT and the TSO optimization engine. So if you apply mid-study changes, those get passed in. So we get whatever changes are going on in the study, we can account for, get a new forecast, and see how that impacts our settings. Okay. And so that's a summary of what trial optimization is and how you can apply it in your clinical trials to have forward-looking information and then how to act on that forward-looking information.
Well, thank you both very much for this insightful presentation. I'd like to, at this time, invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of our webinar. Now, I've already received some questions from folks, so we'll get ourselves started with those. Let's take a look here. Our very first question that I have for you would like to know, well, who enters the data into the system? Is this done by us or Clario?
Well, Tony, I would like you to answer this question.
Sure, thanks, Paul. We have a set of screens that are available for entering, and I believe this question is focusing on the modeling data, the data on the enrollment rates and the different probabilities that we saw for events occurring.
Yes.
So that data can be entered in by either someone at Clario, or customers can be trained to enter that information and then update it as needed. Once the data is all in there and you have your forecasting screens, you can generate a new forecast at any time. So, you know, again, based on the current actuals, so the system will automatically provide you the most recent data. But as far as maintaining that modeling data and reviewing and adjusting it can be done by the customer or by Clario staff.
Excellent. Thank you so much. Now, I believe the next question I have here might be directed towards you, Tony. And I think you addressed this mostly, but I'll ask it anyway. They want to know, does the trigger and resupply model get updated when enrollment is complete?
Yeah, that's very key. That's a good point. As I mentioned, there is a definite time element to that. Once enrollment is complete, for example, we don't want to continue to ship drug to sites for potential new patients. We just want to ensure that we have sufficient stock on hand for the current patients. We don't automatically update those resupply settings, but when the clinical supplies manager views the optimization settings, he or she will see that the recommended floor and ceiling, or the recommended floor and ceiling values are now, they won't be zero because we still have the uncertainty of the randomized active patients getting either 3 or 2 milligrams. But for example, the placebo values could certainly be set low. So as the study progresses and you view those optimizations if the site's completed enrollment or the study's completed enrollment, those numbers will be adjusted accordingly, just so ensure that the current patients have sufficient stock on hand.
Very good. Thank you so much. Our next question here is wondering, are mid-study changes supported?
Yes, they are. Actually, that was one of the reasons for IDDI to select Clario as a technology partner, is because mid-study changes were possible. As you have seen in the slide that I showed, mid-study changes are really considered at IDDI as a partner activity between the biostats team and the RTSM team, just to make sure that we implement the correct changes, and using the Clario RTSM system that is very flexible, that allows these mid-study changes, that is really an advantage for us and for the client. Tony, I don't know if you want to add something to that.
Yeah, I think we want to move to a model where mid-study changes are not an exception, but that they're just a standard part of any clinical study. As we move more, especially in the case that Paul gave, as the study progresses and we get a hold of additional information that we want to leverage in the study, we want to make it easy, or at least have a standard ways of handling it so that we can adjust the trial or adapt the trial, I think would be the right word, to adapt the trial based on that information. So yes, the study model can be updated. Great example of adding a new stratification factor and backfilling those values for the existing subjects. The minimization randomization adjusts for that. And same with the trial supply model. So there's an integration, as I mentioned, between the IRT and our optimization engine. So you don't have to do anything you know, the mid-study changes are automatically applied. Now you might have to make some modeling assumptions now that we have an additional stratification factor or treatment arm. Some of those percentages that have dependencies on that would have to be updated. But other than that, it's a routine process, and you can leverage that to, you know, have it implemented on, you know, relatively quickly and smoothly, and then make, you know, adapt your trial to make use of both in terms of the statistical side as well as optimizing the trial supply in response to that.
Wonderful. Thank you so much for that very detailed answer for that question. The next question I have here is in regards to titration. It wants to know, can the system accommodate titrating up and down multiple levels, such as from 5 milligrams down to 2 milligrams, and accommodate or restrict multiple titrations? And further, is stepping up or down multiple times an option?
Yes, it is. And again, this is really a configuration in the Clario ERT system. So maybe, Tony, I will let you answer that one.
Yeah, we saw in the setup for this particular trial, we only permitted down titration. So patients on 5 could go to 5 or 3, but you know, you could have like for example, a pain study where initially patients may not be tolerating a certain dose, so you have to step them down for that reason. And then once their tolerance builds up, but they need a higher dose for symptom control, we can up titrate them and then, you know, later visit down if needed, and then back up or down. So it's just a question of what's permitted by the protocol and how that's done. And then that can easily, or that, you know, that can be accounted for in the IRT. We have options for, you know, variable dosing, titration, et cetera. And then for modeling that, we would just need to have some estimate of, you know, how often you think patients will titrate it and at what rate. And then of course you can adjust that as needed over time to account for, you know, if the actuals start coming in looking differently than what you had initially expected.
Right, wonderful, thank you. The next question here is wondering, is the supply setting adjusted depending upon the stockpile of the kit at a site? Will the site manager let us know what is the stockpile before we apply different assumptions when computing the supply quantity? And I think, Tony, this might be something you can answer for us.
I'm not sure I fully grasp it, but let me explain my understanding of it. So in this case, the IRT is managing the entire supply of study medication from shipment to the depot, to the sites, and even if we wanted to, the return. So we always know, or the system always knows the inventory of drug at a site. It knows the you know, the quantity by treatment arm, and a clinical supply manager who's unblinded can see that. So it's going to know real time what the inventory is, and then be able to base all the shipment assumptions based on those current inventory levels. It's also doing things like monitoring expiry date. So if you do have lots that are going to be expiring over the course of the study, it knows when to replace those kits so that in time, so that when the kits that are on hand do expire, the replacement kits have already arrived, and that we're not dispensing kits to subjects that are going to expire during the extended time of the visit. So the IRT knows all that information, and then the resupply parameters will be based on that. It also knows how many subjects are randomized at the site. And, you know, for example, if there's 3 placebo patients, it knows, okay, I have a visit coming up in 2 weeks. I'm going to need to dispense a placebo kit. So even without the supply optimization piece in place, it is forecasting kit demand. We call that a forecasting window based on the subject's randomized treatment group, and will ensure that there's sufficient stock on hand for all those planned upcoming visits. So if it's in this particular case, if it's a 5 milligram patient, it knows, okay, I've got this visit coming up in 2 weeks. I need to make sure there's 1 5 milligram and 1 3 milligram kit available, and does that in advance so that you know, if you just happen to say have 3 patients that were all coming in on the same day, it knows what those windows are, and will ensure that there's sufficient stock on hand to meet that visit schedule.
Wonderful. Thank you so much for that answer, and both of you for all of your answers today. However, we have reached the end of our Q&A portion for the webinar. Now, if we couldn't get to your questions, the team at Clario will follow up with you, or if you have some further questions yourself, you can direct them to the email addresses that are up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues so they may view the video once they've registered for the recording there as well. So I encourage you to do that. Now, I'd like you to please join me in thanking our speakers once more for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you very much for coming.
Thank you.